Thanks for inviting me. It's always a pleasure to be in Brazil. And yes, last year you invited me to give a lecture on these old days, first AIDS patient and HIV discovery, which I enjoyed very much giving. And when this year you offered to redo the same thing, I had the feeling that I cannot just stay a person from the past. I have to still be a person, in the present, maybe for the future. And as you just said, although now most of my work is more focused on autoimmune diseases and development of new therapeutics in autoimmune diseases, I've kept aside a project on HIV vaccine because it's not so easy to really leave the field. And so this project and the results that we have now that I'm going to discuss really was based on first starting to understand, or not to understand, to think about what a good HIV vaccine lacks, or what vaccine in general nowadays, those that are developed, lack when they're not efficient. And in fact, I entered the field through the design of an HCV vaccine, for which I will present some results, because there are actually comparison possible between what you do in the design of an HCV vaccine and for the design of an HIV vaccine. So what we've developed is really a platform of vaccine vectors which are derived from retroviruses. And it's in fact the making up of virus-like particles derived from retroviruses and pseudotyped with various kind of envelopes. So why did we started with this project? In fact, this was before this and many other papers appeared in the literature after the failure of the Merck vaccine that Esper discussed earlier, and which actually make people reopen their eyes to the fact that at least 50 years of studies in vaccinology have proved that if you want to design a good preventive vaccine, you have to induce neutralizing antibodies. And the problem with this is that for most of the vectors, the recombinant vectors that were developed by different teams, they were mostly aimed at inducing T-cell response against defined epitopes or larger chunk of the HIV proteins. I've always asked myself why is that, why despite all this literature saying that neutralizing antibodies are key to the development of an HIV vaccine, and that's also true for other diseases, why do people concentrate on inducing T cells? Of course, T cells, as you know, are required to mount a good B cell response. I think also being myself, in fact, a cellular immunologist, working mostly with T cells, there are always been something more aristocratic about T cells than B cells for some reason that I don't have time to discuss here. All this restriction of the presentation, the complexity of the interaction with the different APCs, etc., etc. So we not to be too long about this introduction, we decided that really we wanted to try develop a novel platform specifically aimed at inducing neutralizing antibody. And because the only virus I knew was a retrovirus, and in fact because at that time I was trying to engineer retroviruses as gene transfer vector for gene therapy purposes, I realized that there was a possibility to in fact use this last 10 years of deep knowledge into how retroviruses can be engineered to become to become gene therapy vectors. It was possible to use all this knowledge to design VLPs that could be an interesting vaccine platform. So again, why VLPs? Here, a slide that is a quick summary of development of vaccines over years. As you know, from the 17th century and until now, a couple of vaccines have been developed based on either inactivated or live attenuated viruses. And these vaccines have been extremely efficient. The problem is that sometimes they are not completely safe, and one good example is the yellow fever vaccine, which every year kills a few people, notably older people. And then, you know, there's need to develop maybe better options for some of these diseases at least. Starting in the 80s with the advent of molecular engineering of vectors, etc., subunit vaccines have started to be developed, but not so successfully, and there are, in fact, very few of such vaccines that are really efficient and actually not for complex pathogens. And finally, the big new thing in vaccine design, you know, seen from my very biased point of view, is in fact the development of virus-like particles, which, you know, have been the real big success in vaccine design since the late 90s. And the first one is actually the vaccines against HBV, for which people, you know, usually, you know, did not realize before that in fact it's a really a VLP-based vaccines, as I shall show you later, and more recently the vaccines against HPV. So what this slide tells you is also that, in fact, those very successful vaccines, whether they are inactivated or live attenuated viruses or the VLPs, they share something in common, which is that they recapitulate, in fact, the structure of a viral particle, either because they are, in fact, viruses that have just been killed, for example, or because they're virus-like particle but with the same structure. And this actually makes it possible to use all these co-development of our immune systems, you know, driven by infections and by the fact that now we have developed many means through antigen cells to, you know, captures and particles and present antigens to actually have good and potent immune responses. Also, and most important, going back to the generation of neutralizing antibodies, on these VLPs, like on these viruses, the envelope proteins that are in fact the target for neutralizing antibodies are presented in their correct conformation, which is actually key for the induction of neutralizing antibodies. Okay, so a bit more about VLPs. VLPs have been generated probably from more than 30 different viruses. They consist sometimes of just one, sometimes more viral protein that assemble into particles and mimic the overall structure of the wild-type particle. So here are examples, and for example, HPV, the vaccine relies just on the production of the L1 protein, but the L1 protein will auto-assemble into a virus-like particles with a real same structure than the wild-type particles. And these are other examples. The good thing about VLPs, again, is that they, in fact, are ordered repetitive structure for antigen displays, and the envelopes are conventionally correct. Also, in these VLPs, you usually don't have genetic materials which make them pretty safe, although in some version you can actually add genetic material. Again, to exemplify this, Cervarix, Gardasil, expression of the L1, self-assembly of a VLP. It induces good cellular and humoral immune responses, which is, again, what you want to do when you want to develop protective vaccines. However, this type of VLPs, they offer actually limited possibilities for engineering and use this as a platform to make vaccines against different viruses. Why? Because, in fact, since the VLP is assembled by a single protein, if you insert even just a small T-cell epitope, you have a risk to change the structure and then prevent the formation of a particle. Also, of course, if you want to express larger a protein, like an envelope protein, there are absolutely no way to do this with this L1 protein. So there is a need to develop actually other platform to accommodate larger proteins and to, in fact, try to generate neutralizing antibodies. So we thought that retroviruses could be good to start making this platform. And the reason is that, first, it has been shown long ago that the sole expression of a GAG protein of a retrovirus is enough to make the transduced cell form a particle. Okay, so just GAG is enough to form a particle, and this has been shown in many mammalian cells, but also in yeast, for example. And if you look here at the structure of HIV, in fact, if you express just GAG and ENV in one single plasmid, and if you transfect this plasmid in a cell, what you will obtain is a VLP where the core is formed by unprocessed GAG and the envelope at the surface. And from the point of view of an antigen-presenting cells, from the point of view of the generation of neutralizing antibody, this is actually potentially a very good antigen. Now, of course, you want to do more than just this. And in fact, retroviruses as a platform for development of vaccine really is very versatile, because you can actually do a lot of engineering to improve your antigen. First thing you can do, you can, for example, easily add epitopes into the envelope to have them expressed at the surface. But you can also fuse to GAG sequences that will be co-expressed with GAG in the particle, and these will become potential good antigens for T cells, a good modality for expressing a T cell epitope. But even more than this, what you can do is you can substitute your HIV envelope or any other retroviral envelope for any other protein that you want to express at the surface. And this protein could be either envelopes from other viruses, or even any kind of surface molecule that you would want to address at the surface of the particle to have an adjuvant effect. And again, this has been exemplified, the capacity to pseudotype particles with heterologous envelope has been extensively demonstrated in the field of gene transfer, where in fact, and if you're using lentiviral vector, for example, for your experiment, you know that you pseudotype these lentiviral vectors with VSVG, because VSVG is a very stable envelope protein and allows infection of any cell types. And in fact, there are a series of envelopes that have been pseudotyped efficiently onto retroviral particles, HANA from flu, of course, HIV protein, but also E1, E2 from HCV, et cetera, et cetera. So we believe that this could be a good platform to start work with. Again, to illustrate what I just said, if you express such a very simple plasmid, GAG and ENV, into, let's say here, 293 T cells, you can nicely see the virus bud from the cells, the particle being formed and being released, and they will actually keep here this shape because since you're not expressing the proteases, the core protein are not going to mature, and they will retain this form here. Now, when you have these very simple plasmid, how can you use them to immunize? There are two ways of doing it. I mean, there are maybe more, but the two main ways to do it is first to just use it to produce ex vivo particles, purify the particle, and then use these particles as immunogens. Or you can, in fact, decide to directly inject these DNAs, these plasmids, into the animal. And into the animal, the transduced cells, the cell that will be transduced will actually behave like this one. They will produce particles. So it will be an in vivo production of a particle. So compared to a classical DNA vaccine that will only, probably express, a recombinant protein in an isolated fashion, here, by the co-expression of the two genes, you will produce particles in vivo. Now, when we started the project, we decided to prove the concept with a different antigen. I don't have time to discuss this, but this was published. We showed that we could induce very efficient immune responses with these VLPs, and we also showed that we could develop efficient neutralizing antibodies in the model systems that we used. Now, then we decided for a target to start our real experiment. And in fact, we chose HCV and not HIV to start with. I will show you results with HIV thereafter, for one reason, which is that when you develop a vaccine, a novel platform, if you have good results in your preclinical results, you will then have to go to the clinic, and injecting a novel type of vaccine vector in healthy individual requires a lot of tox studies and expensive studies, and it's in fact much easier to start vaccinate already infected person and try to develop your platform as a therapeutic vaccine rather than a preventive vaccine. So with this reasoning, we decide that maybe it would be smarter to start with HCV, because contrary to HIV, where to my knowledge there is not a single case of one patient who actually was infected and totally eradicated his virus through an immune response. There's one such case which actually occurred after bone marrow transplantation, but so there's no natural eradication of the virus, while in HCV, as you know, at least 30% of the individuals who actually are infected by HCV develop an immune response that eradicates the virus. So there is the intrinsic capacity to eradicate HCV, and in those who actually cannot do so and become clinically infected, it's possible that reinforcing the immune response and actually making them produce the good antibodies, for example, neutralizing antibody, could actually help. So this is why we started with HCV. So again, here an example of just what I've just said. For long in HCV, people realize that almost like in HIV, those person who actually can clear the virus upon infection are those who develop pretty rapidly CD4 and CD8 T-cell response of a significant magnitude. And as in HIV, as you saw this morning, the development of a T-cell response corresponds to, in fact, the disappearance of the virus in the peripheral blood. In those patients who, in fact, have a persistent infection, these T-cell responses are of a lower magnitude. For a long time, people, felt that, the T-cell response is what's doing the job here, and that they should try design vaccine to boost the T-cell response. And the reason for which you were thinking this is because for a long time, there was no test to measure HCV neutralizing antibodies that were available. And when this test, because it was not possible to grow HCV in culture and develop plaque assay or something of that kind. But finally, when an assay became available and people started to look at the neutralizing immune responses in these patients, they realized that also coincident to the eradication of the virus was the appearance of a rapid neutralizing responses, while in the patient with a persistent infection, these antibody actually appeared much later on. So from these results and many others, many reviews appeared saying that while most development programs have been based on improving HCV cellular immunity, current knowledge suggests that it is desirable to include in the same vaccine formulation immunogens capable of inducing both humoral neutralizing cellular responses and of a broad spectrum. And here I think that you can just change HCV for HIV and it will still be correct. So what did we do? As I described earlier, so we engineered two types of particles. One is a particle where we express E1, E2 at the surface of the VLP. Another one would be the expression of E1 alone. You cannot express E2 alone because E1 serves as a kind of chaperone for the expression of E2. And without E1, you cannot express E2. So we could not make the third type of particle, the E2 alone particle. And the reason for which we did this is because we had, from reading the literature, and I cannot really go into the details here, we had the impression that triggering antibodies against E1 would be important for the generation of broadly neutralizing antibodies. Now, again, we made simple construct of that type. We prepared particles. We immunoprecipitated the particles and showed that we could detect both the E1 and E2 protein together with GAG. So particles are made. We also did immunoelectron microscopy to verify the expression of the protein onto the particles. And these are, in fact, images. And there are very few dots here, but even when you do electron microscopy on HCV particles, you don't see much. So it's really pretty satisfying as a staining. And we started to work with this. I'm sorry, the font was changed here. It's not me who chose this pretty weird font here. So it's a Mac to PC transfer problem. So we started to work with these, and we immediately compared the two modalities of immunization that I just described before. One modality is the injection of plasmid to have an in vivo production of a particle. And other is the injection of a VLP that have been produced ex vivo. And in fact, we did experiment where we compared different regimen. Some where VLPs alone were used, plasmid at the end, or some, I'm sorry, actually it's wrong here because it starts with anyhow. So yeah, it starts with one plasmid injection. And there's a group that has only VLP here. So sorry, if it's a mistake, just look at the results here. So we immunized the animals on 3 occasions, 4 weeks apart, and checked here the T cell response against E1 and E2, which are in this construct the sole antigen we're looking at. As you can see, the VLP alone do not do much, and if you don't use adjuvant, VLP alone are not great at inducing T cell responses. But if you start with one injection of a plasmid and then the VLPs, you get better response. And in fact, the more plasmid you inject, the better are the T cell responses. So clearly, for inducing T cell response against the envelope, plasmids are better than VLPs. But again, T cells is not really exactly what we are interested in. We wanted to see the antibody response, and here against E1, E2, and in the end, neutralizing antibody. As you can see here, the more VLP you give after a good prime, if you inject only VLP, you don't have many anti-E1 nor anti-E2 antibodies, and the reason is you don't have a good enough T cell response, and B cell response require a T cell response. But as soon as you get a significant T cell response, when you boost with VLPs, you get better titer. And this is true for E1 and for E2. And our preferred scheme here is to use 2 plasmid injections followed by 1 VLP injection. And it's also where you get the best neutralizing activity for this type of immunization. So we were pretty satisfied with this, and this was the basis for experiment in macaque that I will describe later. We also did a few more experiments in mice to try and understand how good are the VLPs as immunogens, and you will see that they have very specific features. So here in this experiment, we tried to use, since we knew that good T cell responses are extremely important to allow the generation of B cell responses and neutralizing antibodies, we analyzed whether we could have a prime with vectors that are known to really induce very good T cell responses. We started here with an adenovirus that codes for E1, E2. And as you can see, this is a control adenovirus, codes for only GAG-POL, so you have no E1 or no E2 antibody response. But interestingly, when you inject these vectors here and look at the anti-E1 response, you cannot detect any anti-E1 response. You can only detect anti-E2 response. So the first strange result here is that despite the expression of E1, there is no antibody to E1. There are no antibody to E1 that are generated. Now we then boosted these 2 groups here with VLP E1, E2, or VLP E1 alone. And the interesting thing is, of course, you know, when you boost with the E1, E2, you in fact boost the expression of the E2 antibodies. And when you boost with the VLP E1, you do not boost much, but a little bit the anti-E2 response for reasons we don't know. But more importantly, you can actually generate an anti-E1 response. So the nice thing here is that what it shows is that you really, the particle, the E1 particles, have the capacity to generate immune response against protein that, although they are expressed on this vector here, or although they are expressed on this type of VLPs, they cannot be triggered by these vectors. So it's a unique property of this VLP E1 to trigger anti-E1 response. And this we saw in a second experiment, where in that case we use a measles vector to express E1. And measles are replication competent vector attenuated, that's a Schwartz strain. And we use them in strain of mice where, you know, that they can replicate the virus. And this vector was selected because it really produces a ton of soluble E1. And we wanted to first immunize with this vector and then boost. And as you can see, the strange thing is that when you immunize with a measles vector making secretable E1, you don't get any anti-E1 antibodies, despite the production of a large amount of protein. And same thing in this group. And when you boost with the particles, you in fact see the appearance of these antibodies. By the way, I can tell you here that we can detect very good T-cell response against E1. So E1 is expressed, and there is an immune response to E1, but no antibodies. And by the way, here, this slide is also to demonstrate that we engineered E1 to in fact change the tail to express it better on the particle. And when you do this, you have even better immune responses. So the results here are interesting because they show that there is no induction of anti-1 antibodies by a recombinant measles virus expressing soluble E1. And that, in fact, these antibodies can only be generated if you then, you know, boost with VLPs. Okay. Before I move to a macaque study and then to HIV, let me tell you, as I told you before, that there is room for engineering epitopes fused to GAG to generate T-cell responses. We did this also in HCV. And as you can see here, we fused NS3 to GAG and immunized, and we actually got very good and very strong immune response against a GAG against NS3 in this setting. And by the way, this did not prevent the development of good T-cell responses against E1, E2. Okay, so with these results, we went to do a macaque study. Unfortunately, as you know, in HCV, only humans and chimpanzees are susceptible to infection. So there are no challenge studies that can be performed with macaque, but you can at least look at the results of your immunization. So we did a study here that started with a good priming with adenoviruses expressing E1, E2, and then boost by either VLP or plasmo-VLP. And I must say we made a mixture of VLP E1 and VLP E1, E2 injected at different sites, or plasmid coding for E1 or E1, E2 also injected at different sites. Results are summarized on this slide here with a VLP. So let's first follow the blue lines, which show the antibody response against E1 and E2. E1 is actually the dark line here. And as we observed in mice, when you inject an adenovirus coding for E1, E2, you don't produce any anti-E1 antibody while you produce very good anti-E2 antibody response. Okay, so again, when you co-express E1, E2 for some reason, you don't generate an immune response against E1, maybe because the epitopes to which these antibodies bind are masked or not expressed in their proper conformation, we don't know. Then when you boost with these mixtures of VLPs, you in fact boost the titer on the anti-E2 response, and thanks to this VLP E1G, you finally see the appearance of high titers of anti-E1 antibodies. Okay? The T-cell response are in green. They are also generated by the adeno at high level and maintained by the injection of the particles. And there is almost no neutralization here to start with, and it's only when the E1 response appears that you get a neutralization. And we, in fact, in the other group, where we replaced the VLPs, or the plasmo-VLP, we observed exactly the same kind of results. So again, no anti-E1 response after adeno, and only after plasmo-VLP E1G expression you get some adeno immune response, some anti-E1 immune response. Most importantly, we then looked at the neutralizing antibody that we could detect in these animals and checked whether they had the capacity to neutralize other strains than the one that had been used to immunize, which is in fact the H771A strain, which is in dark here. And as you can see here, although it's at different levels, and these are different animals, those immunized with the VLPs and those with plasmo-VLPs, we have a very consistent response where in fact in most of the animals you had a significant level of cross-neutralization, which of course is something very desirable when you develop a vaccine against viruses that show some variation. Okay, so in conclusion for HCV, we showed that our HCV e-VLPs are powerful immunogens. We could show, I did not show you results here, that through engineering you can even improve them and add a lot of protein at the surface. In our experiment in macaques, we had a prime that is necessary to get a good antibody response, but we feel that moving to a clinical trials in HCV-infected patients, the prime is already there, although there's also some exhaustion that can be discussed, and that we could inject straight the particles of the plasmo-VLPs. We show that the e-VLPs and the plasmid e-VLPs induce humoral responses of similar magnitude, that the induction of E1 and or E2 antibodies can be steered depending on the engineering of the particles that you're having. We induce cross-neutralizing antibodies and also good cellular immune responses. So we now are pushing this into clinical development, which is not easy in the case of HCV, but that's another story. And then we want to apply this to other targets, including HIV. And now it's time to enter HIV, as I promised. So what we do for HIV is more or less the same that we did for HCV. We are engineering plasmids to express GAG plus or minus T-cell epitopes, and then HIV envelope protein. This will result in a structure like this, just GAG plus the antigen fused to them, and here we have a collaboration with Edesio to put these CD4 epitopes. And as envelope, there is of course numerous possible choice, and we have here a collaboration with NCD at IAVI to use the most interesting envelopes in these studies. What we did so far, for example, with IAVI, it's a study that convinced Hansi that this was worth pursuing. We did through actually a contracted company immunization in rabbits, comparing immunization with classic DNA vaccines expressing the envelope of GRFL, and then a boost with protein, in that case GRFL trimers. And we compared this immunization and plus or minus adjuvant. We compared this to the response to a plasmo-VLP and then VLP. So it's 2 DNA injection and then 2 VLP injection, or 2 DNA injection and 2 protein injection. And here we're looking at the antibody response. As you can see here, in this combination, as soon as you have a VLP in the and the plasmo-VLP in your combination, and even without adjuvant, you trigger a very rapid immune response of a greater magnitude than when you, in fact, inject just DNA, regular DNA, and then the protein. In fact, while here with DNA alone, you already get a very good antibody response. Here, with this vaccination, you really need to inject the protein to boost an antibody response, which stays of lower magnitude. And even when you add adjuvant to this group, you have a better immune response, but not yet of a magnitude as good as the one that you're getting with plasmo-VLPs and VLPs. So this is encouraging, and we're now working on improving the particles to continue this work. Now, the second thing I want to discuss, where we've put some efforts, is related to the production of IgA, although in fact Esper ruined my presentation here by telling you a bit earlier that IgA are bad for protection. So, I don't know, maybe I should stop here and not look like a fool. But anyhow, no, it's still debated, and we don't know whether IgA are good or bad. But, again, from the literature, you would think that having high enough concentration of IgA locally at mucosal entry site would be a good thing for a vaccine. So what we did is try to study whether by the injection of a VLPs or plasmid coding for VLPs through, in fact, the mucosal route, and we tested the nasal, vaginal, and anal route. I'm going only to show you the results about the nasal route here, which are actually the best results. We thought that maybe by the production of the particles, we could, favor the production of these IgAs that will then, be present locally and hopefully block infection. So before going to the hard target, which is HIV, we used an easier target, which is flu. And what we showed is that by making these flu VLPs, and first by in the injection of DNA intranasally, we could find one molecule, PEI, that was actually inducing a pretty good local immune response through DNA administration. And from this on, we showed that we were able to generate anti-flu IgA. So with this result at hand, we started experiment in HIV. So here is one typical experiment. We inject our plasmo-VLP into PEI, or as a control, we inject regular DNA encoding the envelope protein. We do 3 intranasal injections, and then we look at T cell responses and then at B cell responses by ELISpot or intracellular staining. Results are here and are pretty interesting, I think. If you look in the spleen, for example, in terms of ELISpot, clearly the regular DNA immunization gives you some response, but little, and the response with the plasmo-VLP are actually much better. It's also true if you look in the lung, and if you look at intracellular staining for interferon gamma production in CD4 and CD8 cells, in the lung it's also much better with the plasmo-VLP than with the regular DNA. Now, this is for the T cell response. What about the B cell response? Same experiment, and we in fact looked at IgG and IgA both in the serum, in bronchoalveolar lavage, and vaginal and fecal washes. And we did this by ELISA. Here we're first looking at the IgG, and as you can see, both in the serum, in the bronchoalveolar lavage, and in the vaginal washes, there is a much stronger humoral response detected than when you inject the regular plasmid coding only for the envelope, while here, again, you have this GAG envelope construct that makes the VLPs. So I think this is convincingly showing that plasmo-VLPs are better than regular DNA vaccines for the induction not only of T cell responses, but also B cell responses. Now, what about IgA? In the serum, we could detect higher levels of IgAs after vaccination with this plasmo-VLP injected through nasal injections, and in fact no IgAs after regular DNA injection. And same thing in the bronchoalveolar lavage, you also have higher titers than with the regular DNA, but there is really very little here in the vaginal washes. So intranasal plasmo-VLP vaccines can generate good systemic and local humoral responses with a high rate of specific IgG in the sera, but very few IgG and no IgA in vaginal washes, which actually is not so good. So we decided, in fact, to then do some boosting in this experiment by injecting the VLPs. And to make the story short, when you boost with VLPs here after 3 injections of DNA, in fact, you can boost the immune response, both, in fact, of those animals that had been injected with the plasmo-VLP, and you boost it to high level. But interestingly, you can also boost the response of the animal that had received the regular DNA vaccine to almost not, still a lower level, but still a good response. And you boost not only the IgG, but also the AGA titers. And here it's in the serum, so nothing really special here. Oops, sorry. So the VLP boost enhanced systemic humoral responses, and notably IgA responses. But again, when we looked then in vaginal washes and the feces washes also, we could detect an IgE response that is, of course, of a much lower magnitude than what you saw earlier, and also an IgA response, but still of a very low magnitude. And same thing here. So VLP boost local immune responses, but the concentration of IgA in mucosal washes remains low, and there is need for improvement here. So as a conclusion for this part, mucosal DNA vaccination gives, we believe, promising results after intranasal injection with good cellular and neural responses that are triggered. HIV plasmo-VLP are much better immunogen than regular DNA vaccine expressing GP140. And finally, the boost with the HIV VLP enhances humoral responses, and particularly the local IgA responses. But there still needs for some improvement here. And with this, the general conclusion for our platform, I believe that the so-called e-VLPs and plasmo-VLPs have unique properties that make them good, a good platform for the development of vaccines. They have the capacity to trigger immune response that other antigens cannot trigger, and this is, for example, highlighted by the fact that our VLPs covered with E1 of HCV are the only immunogens capable of triggering good anti-E1 immune response. They present envelopes protein in a conventionally correct manner, and likewise are optimal for the induction of neutralizing antibodies. They are very versatile in terms of design and modalities of use. And I think in conclusion, they present a unique vaccine platform with a wide potential for application. And with this, I will end by thanking my collaborators. So this work has been really done in collaboration with Bertrand Bellier, who was one of my former PhD students and with whom now I'm really developing all of these vaccine projects. The work on HCV has been done by Christophe Huret and Delphine Desjardins. The work on HIV has been done by Léa Torrieri. Other people from the lab were involved in these studies. And we, of course, have external collaborators. We used to work with a small biotech company called Epixis. They've been actually, they generated a lot of the HCV construct. They were also responsible for the macaque studies. And this company, unfortunately, does not exist any longer. It has been bought by an American company, which is now developing this platform to produce an anti-CMV vaccine, and they have actually great results. Macaque studies were done with Roger Legrand and others. The work was done in collaboration with François Cosset and Frédéric Tanguy team, and we have collaboration in New York, Sao Paulo, and also I didn't mention this in Cordoba with Hugo Lujan, where we're trying to make these VLPs suitable for an oral administration. With this, thank you very much for your attention.